Hello, I'm Sandy Deans, the Director of GenQA, and I'm delighted to welcome you to the Genomics Quality Assessment Focus on Preimplantation Genetic Testing webinar. We've got a very busy agenda today with presentations from 4 key members of the GenQA PGT Specialist Advisory Group. So a big thank you to our presenters for taking time to join this webinar today. So before we start, as we've got quite a few people attending on the call, please can I ask you to mute yourself, and if you wish to ask any questions, then please type them into the questions box and we'll address them during the question and answer session after the 4 presentations. So I'll now pass you over to my colleague Mark Sales, who's the Deputy Director for Cytogenomics within GenQA, and he will introduce our speakers as we move forward. Thank you, Mark.
Hello, everybody. I'm Mark Sales. I'm one of the Cytogenomic Deputy Directors for GenQA, and we have several speakers for you to listen to today. These are Dr. Pamela Rennick, who has been part of the Validation and Implementation Committee from the onset of the PGT EQAs in 2008 and continues to act as an assessor for that scheme. She's also the chair of the PGT EQA Special Advisory Group. The second speaker is Dr. Anil Biricik. He has been heavily involved in the delivery and assessment of the PGT EQAs. He has oversight of the delivery of the cell preparation for the PGT EQAs to mimic embryo testing and acts as an assessor for the PGT PGT-A and PGT-SR EQAs. The third speaker will be Dr. Edith Cowan. She's been instrumental in providing scientific guidance for the delivery of the PGTFISH, PGT-A and PGT-SR EQAs, and she also provides an important link between GenQA and the production of the current PGT guidelines that promote good practice and ultimately optimal patient care. The fourth speaker is Dr. Martine de Rijk. She's been involved in the development, validation and delivery of the PGT EQAs over many years and continues to provide her expertise as an assessor. Martine also enabled GenQA's experience in providing assessments for over 10 years to be fed into the current PGT guidelines. This is an overview of the webinar. So Pamela Rennick will cover 10 years of provision of PGT EQAs. Anil will cover PGT-A mosaicism and setting the thresholds for detecting it. Edith will cover 10 years of PGT for blastomia fish, and Martine will cover PGT-M. And at the end, there'll be a live question and answer session. So if you can put your questions into the box for that, that would be great. So the first speaker is Dr. Pamela Rennick.
Hello, everybody. I'm going to just start this off with introducing the different EQA schemes we've been running for the PGT over the years. That didn't move on. Oh, there it's moved on. Right, just to say, why do we do EQA? The EQA is an external validation of the quality of your service according to international standards. So once you've participated in EQA, you will then be given at the end of it an external measurement of the standard of your service. And we all know with this that this is important nowadays for people who are going to be the users of your service, and also people who are being commissioners and funding of your service. So EQA has quite a pivotal role nowadays. As well as giving you this benchmark of the quality of your service, the EQA is always there as a means to improve the quality of the service. And in the schemes, we look at analysis and interpretation. And so for each laboratory, for its performance, you get a feedback on your analysis or your genotype, on your clinical interpretation, and on the clerical accuracy also of the reports that are the final part of this. I think that's still timing through. And also with EQA, anyone from any sort of country can join in with these. So it is promoting internationally quality of services. And it also does have educational aspects to this, because in the schemes we will introduce interesting clinical cases when we have those, and also we have been running over the years various workshops. Presently, we run 5 different EQA schemes for the pre-implantation genetic testing. We have the scheme for the monogenics that has been running for the last 10 years. We also have then the PGTA for the aneuploid screening that's been running, and we also now have the PGT for structural rearrangements, and those are the 3 with the guidelines. Sorry, these slides are still timing through. And yes, those are the 3 with the definitions of the PGT, but we also have kept going on these sort of ones which are the technical specialities. So we also have the scheme for the PGT for the blastomere fish, and we have the scheme for the running for the polar body analysis, which are important parts for some of the PGT centres. Right, so each of these schemes, they get delivered in different ways. The blastomere fish that has been running, that's essentially still like a dry scheme where it has a theoretical workup, and then it moves on to part 2 where there's analysis of online fish images which from historical cases. The polar body scheme was running on a similar way with this. And then we also had starting up, sorry about the slides, with the monogenics service. We did this actually as a completely wet scheme. So this actually does follow through as you would do for a clinical case where genomic DNA is sent out for the workup for a couple for PGD, and then the part 2 we sent out single cells to mimic embryos. The bit that's happened with this, the whole of the schemes for the PGT, is that we had this big the technology, the milestones and everything changed. So we had everything came together technology-wise with a whole genome amplification, and that then allowed microarrays and next generation sequencing to be used. And also around the same time as our embryology-wise, we also had the move to trophectodome biopsy and the vitrification. So we've realised that the platforms were changing. So even though these technologies were around, they kind of got introduced into the PGT areas kind of in about 2012, 2013. So you can see here that we then did pilots of trying and sending out for the aneuploid screening some cells. And then it then changed to actually to sending out genomic DNA. So these schemes came in with the genomic DNA going out, and then also the polar body ones switched to sending out whole genome amplified DNA from actual cases. So that switched from being a dry scheme to a wet scheme. And then our most recent one has been the structural rearrangements. So the participation over the years has expanded year on year. 10 years ago in 2010, there was, I think, 52 labs enrolled. And then the year that's just gone by, we've had over 216 labs enrolled in the different schemes. The bit just to point out with this is that for the monogenics, we change around what disease we are offering. So over the years, we have run autosomal recessives, we've run dominants, and we've run some X-links. So we would try to put out different core diseases. So the uptake of these over reflects the labs of who has a test set up for which of these diseases. So just the comparison of over the 10 years is 10 years ago, there was 3 schemes running, which is the blastomere fish, the polar bodies, and the monogenics had started up just on single cells. And then now, this year that's just passed, we were doing 4 times the amount of work, and here 2/3 of the scheme work combined is the aneuploid and the structural rearrangements. It's not that structural rearrangements is that much of the cases going on, but I think it's to do with the platforms that laboratories are using, and that the labs that have the platforms to do with the aneuploidy will also be able to detect the structural rearrangements. So over the years, we've had to recruit in and have a lot more assessors on for each of the schemes for the way that they've been expanding. I'd mentioned before we have always thought about an educational role with this, and there's been participants meetings which have been held at the annual ESHRA meetings, and that's been since 2009. When we had the change in technologies going on back in 2012, 2013, we held a best practice workshop on microarrays. That was in Athens. And then we also followed up and we had a workshop in Maastricht in 2016 on the impact of next generation sequencing. And there was also a participants update at a meeting in Brussels in 2017. We had, when we first set up the monogenics, when we were doing the pilot and setting that up, we published a paper back then on the 3 years of delivering the service. Quite a few of those people are still on the specialist advisory group, which I'm pleased to say. And then I've just giving a little snapshot here of 4 of the schemes for the last few years of how people have been performing, because I know people do the scheme and you may well then get back that you've got the satisfactory marks from this, which is great. That's what this should be doing. But just to show you here that the standards are maintained here through the schemes, and with this, in each year that there will be a few possible poor performers, and they're ones in this who will then have had advice and feedback with things. So all in all, I think the scheme does a good job. So I hope this has introduced what we've been doing over the 10 years with this EQA scheme, and now we're going to move on to the other sections for the feedback on last year's schemes. Thank you.
Hello everyone, sorry about that. We were having technical difficulties with the transitioning. I think I've managed to fix it now. So I'd like to introduce our second speaker. This is Dr. Anil Biricek, and he will be presenting on PGTA mosaicism and setting thresholds.
Hi everybody. During the recent aneuploidy EQA schemes, some participant laboratories showed poor performance for analysis and interpretation of mosaic samples. So my speech will be on this issue. Okay. And currently there is no any standard guideline to deal with the mosaic embryos, but in 2016, PGTFISH and 2017, CoGen published their position statement to guide laboratories and physicians. And today, suggested thresholds for mosaicism is between 20 and 80%, but some laboratories prefer their own threshold levels like 30 and 70%, or some of them are using less than 50 or more than 50% of mosaicism as threshold. And since the first publication of clinical outcome after transfer of mosaic embryos in 2015, many other data showed the implantation potential of mosaic embryos, and these data showed a pregnancy rate between 15 and 41%. And then in 2017, one mosaic sample has been included in PGT EQA scheme, and it was a mosaic gain of chromosome 7, about 60% of mosaicism. And when we look at the performance of the participant laboratories, 71% of the laboratories have been correctly reported this mosaicism, while 27% of the laboratories failed to report this mosaicism, and they reported all as a full trisomy of chromosome 8, and one laboratory has reported totally incorrect results. And when we look at the platforms used by these failed laboratories, there were NGS, RHGH, and qPCR platforms. And next year, in 2018, again, another mosaic sample has been included in EQA scheme, and this was a mosaic gain of chromosome 7. And this year, the performance of the participant laboratories were better, and 95% of them have been correctly reported this mosaicism, while 3 laboratories failed. And the platforms used in this year between the failed laboratories are NGS and RACGH platforms. And during the EQA scheme of the last year, there were no any mosaic samples. And the samples included were trisomy of chromosome 18, 21, and 16, respectively. But in this year, we faced with another problem because some laboratories incorrectly reported this full chromosome gain as a mosaic trisomy, and most of them were having were using RACGH platforms. When we look at the data, introduced data, we show that the log-2 ratio of these samples were creating this kind of problem. So for a demonstration, if we can see in this slide, in RACGH platforms, sex mismatch is being used to detect log-2 ratio between reference and sample and reference. And sometimes these thresholds may create different levels, because as we know, during sexual mismatch, the chromosome copy number changes is changing from 1 copy to 2 copy, but instead during the autosomal chromosomal changes, copy number is changing from 2 copies to 3 copies, and this may affect the sexual mismatch log-2 ratio. And if you have a log-2 ratio for an autosomal trisomy and very near to the threshold, this may create some confusion, and especially if your sexual mismatch log-2 ratio is less than you expected, then as you can see, your full trisomy may enter an mosaic interval. So this was the problem we faced with some laboratories or participant laboratories of this year. And if we look at the mosaicism performance between RACGH and NGS, we can definitely see that today NGS platforms are much more sensible for mosaicism detection, because RACGH is a comparative method, so labeling of the test and reference samples and log-2 ratio levels may change the performance of an RACGH platform. And as we can see in this sample, if you have a monosomy with the same level of your sex mismatch level for one chromosome, and while the other chromosome have lower log-2 ratio, and then you can again incorrectly consider this monosomy in this slide like chromosome 7, like a monosomy, mosaic monosomy. But actually, when we compare the same sample with NGS data, you can see that the same level of monosomy for this chromosome 7 and chromosome 18, and because in NGS you can see directly copy number, read numbers of chromosomal gains and losses. And of course, also for the low level of mosaicism, as you can see in the slide, like chromosome 19 may show a very small amount of mosaicism levels by NGS method. As a conclusion, actually, mosaicism is a very deep issue, so there are many things to discuss, but I would like to just summarize the problems we showed, we faced with the participant laboratories. As we know, NGS is much more sensitive than RACGH for low level of mosaicism detection, but both techniques have limits for mosaicism detection. And log2 ratio is an sexual mismatch can be used to set log2 ratio for RACGH platforms. And when you see a mosaic sample, this interpretation should be done with care. And even in NGS platforms, sometimes normalization algorithms may affect the mosaicism detection level, and this may cause false positive and false negative mosaic results. And today, there are different commercial NGS platforms, and each platform may have its own normalization algorithm, and some platforms are giving also these filter options to get these the thresholds manually. So some operators can also change these thresholds by manually, but some NGS platforms are set up as a default mode, so you cannot change manually. But in each kind of platforms, array or NGS, it is always suggested to check aeroplate and haploid cell mixing experiments to check the quantification and limits of detection levels for mosaicism. And whichever technology is being used in PGTA, mosaicism detection limits should be given in the report for the correct genetic constant of the patients. Thank you very much.
Thank you very much, Anil, for a very interesting talk and quite an important concept. So our next speaker is Dr. Edith Cowan, who will speak on 10 years of PGT for blastomerefish. Welcome, Edith.
Thank you, Mark, and welcome everyone. Welcome from Maastricht, and thank you for listening. I'm trying to switch over the slides. Yeah. Okay. Okay, so as Pam already indicated, that the PGTFISH for blastomeres has been around in EQA from the start. So we have celebrated, so to say, our 10 years anniversary. And as you can see, that the number of participants has gradually decreased over the years, and that is, of course, because of the introduction of comprehensive methods as RACGH and NGS, taking over the analysis for PGTA and PGT-SR. And as you can see here and in the right lower panel, is that the performance of the participants varies over the years, and that is related to the complexity level of the indications that we selected for PGT fish. So all those years, every EQA consists of 2 cases, and we try to have a variation in the PGT indications. So we had, over the years, we had Robertsonian translocations, reciprocal translocations. We once tried a complex rearrangement that comprised 3 different chromosomes, but found out that that was a bit too difficult. So that was a one-time experiment, but we also included other structural abnormalities like deletions or inversions. So over those 10 years, as I already indicated, we had a change of purpose. As you can see here in the very beginning in 2010, more than half of all the participants were for the blastomere fish, 58%. So at that point in time, it was the sole technique that was available for the cytogenetic indications. Whereas now, as you can see in the 2019 scheme, it is only 5%. So now it's sporadically used technique that is in most centers only used to detect small imbalances that cannot be detected using the comprehensive techniques. So what we try to do with each and every EQA scheme is make sure we mark against a consensus guideline or against practice recommendations. And for the PGTFISH, until last year, we have been using the ESHRE PGT Consortium best practice guideline for fluorescence in situ hybridization that was published in 2011. So it was high time that their new guidelines came about, and in 2020, so very recently, another set of guidelines was published that we will be using from 2020 onwards as to mark against. And this is the ESHRE PGT Consortium Good Practice Recommendation for the detection of structural and numerical chromosomal aberrations. And I don't know if you will be able to get a copy of the slides, then this is the link to this open access publication. So now on to the 2019 EQA scheme. As always, in part 1, we wanted participants to do a workup of the 2 cases that we provided, given the clinical and referral information. And laboratories were expected to explain the probe strategy that they would use if this were an actual case in their center. And the part 2 consists of an online analysis of FISH results of blastomeres from IVF embryos. So we supply images, FISH images that the participants need to interpret. So the case scenarios for 2019 were a balanced reciprocal translocation between the chromosomes 7 and 8, and the validated results were that all embryos were unbalanced, so there were no embryos suitable for transfer. In case B, we had an individual that was mosaic for a derived chromosome from the translocation 820. And here we had 2 embryos available for genetically available for transfer, which were the embryos 3 and 6, and they were normal/balanced, as on an interface level we cannot see the difference between the 2. And then we always try to have our images as straightforward as possible, but last year we decided to withdraw 1 embryo from the analysis, as it turned out that the orange signal was considered inadequate. So how did the participants perform? We had 11 participants enrolled, and 10 of them had a satisfactory performance, which was quite good. Unfortunately, 1 laboratory received a poor performance because for both cases they had a critical interpretation error. So the PGTFISH EQA is marked on 3 different levels. In green, you can see the analytical part. So do you have the results right? The interpretation part, which wants you to explain what this abnormality you found what is the meaning of that, and then the clerical accuracy. And as you can see, most mistakes are always made on the interpretation level. And last year we had a rather complex case as a number of laboratories failed on the interpretation part and lost points there. So what comments did we have for this EQA scheme, the 2019 scheme? That, well, the purpose of the assessment is always to examine the service provided by to the patient, and not just the technical abilities of the laboratory. So we want a genomic report that is good both for the provider and for the patient, and a well-performed interpretation of the results. So in case A, we had a balanced translocation with a small segment exchange. We had one lab with a critical interpretation error because they selected an unbalanced embryo for transfer. And an important point in this case A was that the breakpoints were so telomeric that participants had to be aware of the fact that independent segregation should be considered, as well as quadrivalent formation. So for case B, we had a parent who was mosaic for a derived chromosome. Again, we had one critical interpretation error, as an unbalanced embryo was selected for transfer. And in this case, we actually only had only two segregation patterns possible, either the normal or the unbalanced for the derivative chromosome 20. And in this case, the reproductive risk figure could be as high as 50%, depending on the distribution of the DAR20 cell line in the ovaries. So what other issues did we see? 5 out of 11 respondents specifically identified the segregation products that I just described, and that were most likely to be viable. That is one thing we always like them to do, explicitly explain which products are most likely to be viable. Sometimes we have labs that take a two-cell biopsy while the probe strategy that they are using is good enough for a single-cell biopsy. And of course, when you don't need 2 cells taken from the embryos, you should consider not doing so. And so the last remark is on the fact that we included an embryo from which a binucleate nucleus was taken. And sometimes this causes problems. For one participant, it did, because it did not recognize that it was a binucleate. And of course, you need to take care how to interpret the results of the 2 different cells, 2 different nuclei. In case B, we had also 5 out of 11 participants that considered the possibility of those 2 segregation products, and while the others incorrectly gave additional segregation patterns for the quadrivalent, because as these imbalances were so small, there is a very low to nil chance that they will form a quadrivalent. Again, 2 labs suggested a 2-cell biopsy where that was not necessary. And here we had we included another issue, because we had an embryo from which 2 different blastomeres were taken, and that is of course on an interpretation level quite different from the binucleate one that we included in case 1. So one laboratory incorrectly stated that the embryo could be transferred, which it couldn't, because we had 2 blastomeres, of which only one was diploid and the other was haploid. So so every participant receives an individual laboratory report, and if they do not agree to the points that have been deducted, they can file an appeal. So in 2019, 2 laboratories did just that. but following consultation with the assessors and with the SAG, both appeals were rejected. So this was my last slide on last year's scheme. I'll be happy to take any questions afterwards and hand over to Mark.
Thank you, Edith, for that really good talk. The next speaker is Martine de Rijk, and she's speaking about PGTM.
Yes, thank you. Good afternoon, everybody. I'm just waiting for the control of the slides. Okay, so in this first slide, you can see a summary of the overall outcome results for the 2019 EQA PGTM for Huntington disease. And in the stage 1 feasibility testing, all 56 participating labs achieved acceptable performance for genotyping, while 2 labs were classified as poor performers in the category of interpretation. For the testing of the cells in stage 2, there were 5 poor performing labs in the category of genotyping, and then 3 in the category of interpretation. So overall, that was well done. Now, which trends did we observe in the 2019 EQA? Well, this graph shows the methods and the samples used by the participating labs in the last 3 EQAs. And for the methods, that is the STR-based testing in green and blue versus the SNP-based testing in yellow and olive green. And when you do the calculation, you see that for the methods, there is actually no change over the years. The STR-based testing is still the most used method. 60 to 70% of participating labs rely on this method. SNP-based testing is applied in 30 to 40% of the labs. Then for the samples, and this is then the single cell day 3 biopsy, so the green and the yellow versus the trophectoderm biopsy at day 5, day 6, blue and olive green. And there you see that there is, for the day 3 biopsy, a decrease. And indeed, so there is a shift towards trophectoderm biopsy. And in the 2019 EQA, 80% of samples were already day 5, day 6 biopsy samples. So now I move to the assessor feedback for the 2019 scheme. And the first item is about the interpretation of the informativity stages of the genetic markers. And that interpretation was inconsistent from lab to lab because different definitions are used. And so in order to come to a more uniform interpretation, we advise that for the next EQA, that labs are using the definitions as given in the recently published ASHRAE good practice recommendations. So another feedback remark is about assuming homozygosity in cell samples. So when testing genomic DNA samples, and when only one allele is detected, the genotype is interpreted as being homozygous. That's okay. However, when testing single or few cells and only one allele is detected, labs often interpret this as that cell being homozygous, but it can't be assumed that two alleles are present for that marker. And this is a point that actually has been raised now for several years. And so it has been decided that from the 2020 EQA scheme onwards, marks will be deducted. So the next feedback is on the whole genome amplification protocol. It is recommended to rely on an MDA-based method. This because there is, for the haplotyping, a better genome coverage, and also the genotyping error rates are much lower with the MDA-based method. So labs that are still opting for a non-MDA-based method, they actually require a full validation assay. And then my final feedback is about SNP-based carrier mapping. It has been recommended to perform a feasibility study before offering embryo testing. Now, as there is no phasing option included in the carrier mapping software, the informativity testing and the phasing should be done manually, and that is feasible in theory, but in practice, many labs relying on carrier mapping do not consider this. And since the software only generates the stage 1 results then after processing the cell samples, the labs can't provide stage 1 results. And to overcome this problem from 2020 onwards, it has been decided that the EQA samples from stage 1, so the genomic DNA samples from stage 1 and the cell samples from stage 2 will be distributed at the same point in time. And this also means that the labs will do the reporting on stage 1 and stage 2 also at the same time. Labs that after testing of the stage 1 samples conclude that they can't proceed to stage 1, well, they simply report then only the stage 1 and they may discard the cell samples of stage 2. So it is actually a big change with all the previous EQA schemes where we really had stage 1 and stage 2 clearly separated from each other. So then in my last slides, I come back to the STR and SNP-based methods to discuss more generally the pros and the cons of these methods. So in this slide, what is shown here is actually that these methods do share the same principle of haplotyping. So whether you now use the SNP markers in a genome-wide approach, or you rely on the STRs in a more targeted approach, there is always the need for informativity and phasing, and also the requirement for the family samples. That is just very similar. What is the difference? Well, with the STR-based methods, there it is a customized test. The consumables are relatively cheap, but here the high cost comes from the personnel cost, as you have to find, develop and validate STRs with every new target. And especially the development and validation at a single cell level is labor intensive with long waiting times for the couple. In the SNP-based testing, so with SNP arrays or NGS, it's different. That specific problem is overcome as now labs mostly can rely on commercial kits with standardized workflows, and these require just a single time implementation validation. The downside here is that the equipment and the consumables are quite expensive. However, samples can be pooled, allowing you to cut down on personnel costs considerably. Another advantage of the SNP-based testing is that it is quite powerful with regards to double indications, because you can then do the diagnosis for more than one disease from just the same data set. And another very positive point is that the data not only yield information on genotype, but also information on chromosome copy number, meaning that for labs wanting to combine PGTM with PGTA, this is the method of choice. So this was my last slide. I would like to thank you very much for listening, and the next part will be on questions.
Thank you very much, Martine, and to all of our presenters on this webinar. I'd also like to thank, on behalf of GenQA and the whole team based in Oxford and Edinburgh, a huge thank you to our PGT Specialist Advisory Group members who are past and present members, the PGT assessors, again past and present members of the assessment teams, also our sample sourcing laboratories and those labs that also very kindly perform our validation testing prior to the EQA distributions, and also to the lab in Rome, Genoma, for the cell tubing for the PGT molecular EQAs, which is a very hands-on, heavy job, and we are entirely grateful to be able to deliver the embryo testing for PGT with their help. So now I'm going to open the floor for questions, and we've had a few questions through the system. So Mark, I think you've got a couple and I've got a couple here. So will I just kick off, and if we pop it onto the next slide, you'll be able to see our panel members who hopefully will be able to address your questions. So the first question, and this is something I know when we've been doing the assessment for the PGT EQAs, often comes up when we discuss this. So for STR analysis, how many flanking markers should be used, and what if one is intragenic? What are the views there? So maybe Pam, will I come to you for that one, please? And then maybe Martine can come in.
Right, yes, and I can do. That's something that hasn't changed over the time. It may be a little bit with the fact you have trophectoderm biopsies now, which have several cells in them. It is that ideally it's markers each side flanking the gene, and they should be close in, so you're not going to get affected by recombination events. So you need to have informative ones, which Martine was talking about, informativity. You need to have informative markers within a megabase either side of the gene with this, but then you can kind of substitute in if you've got an intergenic one as well, but that you don't get so many informative intergenics. So it's still that same rule of thumb of flanking within 1 megabase. Really can't go wrong on that with STRs.
Great, thank you, Pam. Martine, is there anything you want to add?
Well, I would add that there is the minimum number, but I would say the more STRs, good informative STRs that you can add, the more robust your test will be. Because when you just stick to the minimum and you have an ADO or a recombination or a contamination event, you may end up with no diagnosis, whereas in the case where you have more markers available, you you may still reach a diagnosis for that embryo.
Okay, thank you. Very good advice there. Thank you very much. Mark, do we have some aneuploidy or structural rearrangement questions?
Yeah, I have a question for Edith. Can you explain why the PGTFISH now focuses on small segment exchanges rather than larger rearrangements?
Yeah, sure. As I already indicated in one of my slides, that the larger imbalances are very elegantly analyzed by the comprehensive methods that are available right now, especially ARIS-CGH and NGS. So this also gives you information on the ploidy status of the other chromosomes, which is often used by a number of labs. And if you only do FISH, you get only information of the 2 chromosomes that are involved in the indication. So only the very small ones, the comprehensive methods do not have enough information points, so to say. So that's why in those instances you need to rely on the old-fashioned FISH.
Thank you. That's a very good explanation of that.
Okay, Mark, there's a couple of actual EQA questions here.
Okay.
So maybe we could probably do this. Both are PGT molecular. So when the lab is changing their methods, for example, SDRs to karyomapping, should this specifically be mentioned in the submission to GenQA? So the answer to that is yes. You will see for the results submission for stage 1, we require a completed proforma to be submitted and no clinical report. And for stage 2, we're looking for a second proforma and a clinical report to be submitted. And those proformas are different depending if you're doing STR analysis or SNP analysis, as obviously the data that we collect is different. So you'll see there that you have to select which one to submit and complete. And then the second question from the same person is, from 2020 onwards, will the time for submission of the result be the same, and what is the time given to the lab for performance? So the DNAs and the single cells and the cells will be distributed. We're planning for them to go out on Monday next week, and the closing date will be the same, and that will be 8 weeks from submission, from distribution of the samples. So we've extended the testing time to incorporate both stages. Great, thank you. Have you got anything else coming through now, Mark?
Yeah, I've got a question for Anil. Do you feel it's important to do the experiments to set the limits for your own platforms? And which platform would you think is better, whether NGS or ARIS-CGH?
Actually, I don't want to specify any platform which is better than the other, but I think I can just suggest that NGS platforms are currently much more sensible. We know that. So, of course, during the EQA schemes, we showed that some laboratories showed very poor performance despite their NGS platforms. So that's why the main thing is to just do cell mixing experiments before the application of clinical cases. This would be better for the laboratories. That will be my suggestion.
That's great. Thank you for that, Anil. Do you have any other questions, Sandy?
No, nothing coming up here. I would suggest that we close the call now, because we're now on at past. So a big thank you to everyone joining us today. If we go to the next slide, Mark, please, we can give some information on feedback, which would be greatly appreciated. And whilst you're doing that, just a big thank you to our presenters once again. If anyone does have more questions they want us to address, then please feel free to send us an email at info@genqa.org, and we will definitely either answer them within the team at GenQA or pass them on to the assessors. So feedback is very welcome. and a survey will be sent to those of you who have registered following the webinar. The webinar will also be available to view on the GenQA YouTube channel, so please click subscribe and you'll have access to this. We will also issue participant certificates for this webinar, and just a heads up notification that the October focus on topic will be on molecular pathology and NTRK fusions, linked to the pan-cancer drug treatment options which are now available. So once again, thank you very much everyone, and I hope this was a useful session. Goodbye.
Bye.
Bye.
Goodbye.
Goodbye.